In this lecture, we're going to talk about how viruses replicate. While the replication cycle of viruses can vary from virus to virus, there's a general pattern that can be described consisting essentially of 6 steps: attachment, penetration, uncoding, replication, assembly, and release. We'll go through each of these individually. So the first step is attachment. Outside of their host cell, viruses are inert or metabolically inactive. Infection is initiated by a collision between a virus particle and a cell, totally by chance. Therefore, if you increase the concentration of virus, you increase the probability of infection. The attachment itself is highly specific between molecules on the outside of the virus and receptors on the host cell surface. This accounts for the specificity of viruses to only infect a particular cell type or particular host. Target receptor molecules on cell surfaces may be proteins, usually glycoproteins, or the carbohydrate residues present on glycoproteins or glycolipids. Virus particles cannot infect every cell they encounter. Susceptible cells express appropriate receptors. Cell receptors determine the host range and tropism, or predilection, of a virus to invade and replicate in a particular cell type. For example, poliovirus grows in primate cells, but not mouse cells. This is because mouse cells lack the appropriate receptor. This has been demonstrated quite nicely using molecular biological techniques. A small animal model for polio was developed by producing transgenic mice that express the appropriate primate receptor. In this picture, you can see the variety of different kinds of molecules that can act as receptors. So, for instance, polio uses an immunoglobulin superfamily. The loops indicate homologous domains stabilized by interchain disulfide bonds. LDL can be the receptor for some viruses. Sialic acid is a common receptor for influenza virus. Some receptors are transport proteins. But despite all the variety in the types of proteins that can act as receptors, there's one factor that unifies all virus receptors, and that is that they did not evolve and are not manufactured by cells to allow viruses to enter cells. Viruses have subverted molecules required for normal cellular functions. So this is an important point. Keep in mind that it is not to the benefit of a cell to evolve receptors for virus. Instead, the virus has found ways to exploit existing molecules on the surface of a cell that they can then use to enter the host cell. Cell receptors to virus can be classified into 2 classes: adhesion receptors and entry receptors. Adhesion receptors attach the virus in a reversible manner to target cells or organs. This adhesion is not mandatory for virus entry and alone does not trigger entry. However, it enhances infectivity significantly by concentrating the virus in the vicinity of the entry receptors. Entry receptors trigger virus entry by endocytosis or by inducing fusion, and the consequences of this binding is irreversible. Adhesion and entry receptors are often referred to as co-receptors. So let's look at influenza virus as an example. Influenza virus has 2 types of glycoprotein spikes on the envelope. One is the hemagglutinin, or the HA. This particular molecule binds to sialic acid as its receptor. And this attachment can be reversible. The Na cleaves sialic acid from sugar side chains. And it may sound like it's working against the HA, but it serves an important function. The Na cleaves sialic acid so that it can elute viruses that have bound inappropriately, and it also saves the virus from mucus. Remember, influenza virus is a respiratory virus. Our respiratory system has all kinds of protections in it. So one type of protection is we produce mucus within our respiratory system, which, as you all know, is a pretty sticky material. And it's moved from deep down inside the respiratory system, and then cilia that line the respiratory system beat in a way that it moves the mucus out of our respiratory system. As the mucus is moved, it picks up all kinds of particles that we have breathed in. And so it serves a cleaning function for the body. And then once the mucus makes its way up to the top of the respiratory system, we can either swallow it, spit it out, whatever. But it's a good way to clean out materials that could clog up our lungs if we didn't have a cleansing mechanism in place. Now, mucus has a lot of sialic acid on it, and influenza virus particles can get bound up to mucus. The Na will help cleave the virus particle from this, and hence they use the term it saves the virus from the mucus. Many naked viruses inject their nucleic acid into the host cell, leaving an empty capsid on the outside. This process is termed penetration and is common with bacteriophage, the viruses that infect bacteria. With the eukaryotic viruses, it is more likely for the entire capsid to gain entrance into the cell, with the capsid being removed in the cytoplasm. An unenveloped eukaryotic virus often gains entry via endocytosis, as you can see at the bottom of this figure, where the host cell is compelled to engulf the capsid, resulting in an endocytic vesicle. An enveloped eukaryotic virus gains entrance for its nucleocapsid when the viral envelope fuses with the host cell membrane, pushing the nucleocapsid past the cell membrane. If the entire nucleocapsid is brought into the cell, then there is an uncoating process to strip away the capsid and release the viral genome. So you can see here we have receptor-mediated endocytosis, but also with envelope viruses, you see up at the top, the envelope can fuse with the plasma membrane at the surface of the host cell, and then we have a fusion, and the entire nucleocapsid can be brought into the cell that way. This is an example of attachment and penetration by pseudorabies virus. So you can see up in the top left, you have receptor binding, and then in the top right, you have more receptors getting involved in the binding. And then ultimately, down at the bottom right, you can see that you have the fusion of the envelope of the virus with the membrane of the host cell. And the nucleocapsid now has access to the cytoplasm of the host cell. When we look at plant viruses, they enter host cells in a very different way. Remember, plant cells have a tough cell wall, so you need a break in the cell wall for the virus to be able to breach the cell wall and enter the host cell. This is usually accomplished through damage, either by various kinds of cultivating techniques, pruning and so on, or feeding by insects, as you see in this picture here. Once the virus has breached the cell wall, it can use plasmodesmata to move from cell to cell, and it's carried by phloem, so that you get a systemic infection moving throughout the plant. And in this picture here on the right, you can see two individual plant cells side by side. The light gray would be the cell wall, and you can see the channels running through the cell wall, connecting the two different plant cells, and those are the plasmodesmata. So these channels allow for the movement of materials from cell to cell within a plant. The next step in virus replication is uncoating. This is important because, remember, the capsid protects and contains the nucleic acid of the virus particle, but the genome has to be exposed in order to become active once it's inside the host cell. So the capsid has to be completely or partially removed. This often occurs in endosomes and is induced by low pH. Acidification is a really important step in this, and that's why this process often occurs in endosomes, because endosomes will pump protons or hydrogen ions into vesicles using the so-called proton pump. And as the hydrogen ions are moved in, the pH within the endosome drops. And this process has been shown experimentally very nicely by using a drug that prevents acidification by inhibiting proton transport. We won't worry about what the name of the drug is. But when cells are treated with this drug, it completely inhibits infection with a number of viruses, such as influenza. And that's because they all enter the cytoplasm from acidified endosomes. When you treat the cells with this drug and it prevents acidification, the viruses can't be uncoated, and so replication doesn't occur. Most viruses leave the vesicle before lysosome fusion, but this can vary from time to time. So we have two particular examples here. On the right side, we have receptor-mediated endocytic entry of an envelope virus. So the virus is enveloped. First it attaches, then you get the formation of the vesicle, the endocytic vesicle. And inside that endosome, you get acidification because of the proton pump. This has an effect on the capsid of the virus, and then the nucleocapsid is released into the cell's interior. On the left, we have receptor-mediated fusion of an envelope virus with the plasma membrane. So the virus comes along and binds with the appropriate receptors on the surface of the plasma membrane. This process of attachment leads to fusion of viral and cellular envelopes, and then the nucleocapsid is released inside the cell. And the virus can then patch that spot where the virus fused and make the plasma membrane whole so that the virus is able to function. In other words, it doesn't punch a hole into the membrane of the host cell, because that would kill the host cell. Okay, once the nucleocapsid is inside the host cell, then we move on to replication. And one of the best ways to study replication of virus is to use what's called a one-step multiplication curve. And this allows you to study a single replication cycle. And the way you do this is to allow only a short period for absorption, because you want to have the virus synchronized. If you allowed for a long period of absorption, then different virus particles would be at different stages of replication. So you only allow a short period for absorption. You wash off any unattached virions, and then you treat the cells with the receptor-destroying enzyme to prevent infection by released virions. So what this process does is ensure that a single replication cycle occurs synchronously in every infected cell. So that's your one-step multiplication curve. And what we find is that there are various steps. So if you look at this graph, it's a very simple graph. It's just time versus virus count. And at the beginning of the graph, you add the virus. And of course, there's going to be a period of time during which you can't find any infectious virus. And that's because you have the virus has been uncoded, and now we're getting replication of the various parts of the virus. Because remember, the virus is produced in segments, in pieces. So you have replication of early enzymes, you have replication of the nucleic acid, then you have replication of the protein coat. And during this time, you can't detect any complete infectious virus, and this would be the latent period. So this is the time between disappearance of infecting virus and appearance of new virus in the surrounding medium. Now the eclipse period, this is the time when infecting virus is uncoded, so you have no infectivity to be detected using a plaque assay. But the virus is indeed very busy within that host cell. So during that time, though, you can't detect it. Then you have maturation, and then assembly and release. You have what's sometimes referred to as an intracellular accumulation period. And this depends on the type of virus. Does the virus bud from the plasma membrane, in which case the virus doesn't really accumulate within the host cell, it's constantly being released? Or is it really released from the host cell by a lytic process? If that's the case, then you're going to have a significant intracellular accumulation period as virus accumulates within that host cell, and then a lot of virus is going to be released at one time when the cell lyses. And then, of course, we have to look at the type of virus genome. That's going to affect how the virus replicates. We have 4 basic types of virus genomes. We have double-stranded DNA, single-stranded DNA, double-stranded RNA, or single-stranded RNA. The synthesis stage is largely dictated by the type of viral genome. Generally, DNA viruses copy their genomes directly to DNA, and RNA viruses copy their genomes directly to RNA. There are, however, some DNA viruses that replicate their genomes via an RNA intermediate, and some RNA viruses that replicate their genomes via a DNA intermediate. The various replication modes of genomes are summarized in this figure using the Baltimore classification. Viral-specific enzymes such as RNA-dependent RNA polymerases, or a polymerase that makes RNA from RNA, might be necessary for the replication process to proceed. And these kinds of viral-specific enzymes can be very important targets for drug development. So, for instance, the enzyme that makes DNA from RNA during HIV replication was the target of many types of drugs, including AZT. Protein production is tightly controlled to ensure that components are made at the right time in viral development. We'll look at all of these steps in much more detail as we go through the individual virus families. This is just an overview. When viruses infect eukaryotic cells, the genomes of some are delivered to the cytoplasm and some are conveyed to the nucleus. The destination of a virus genome, and hence the location in which it is replicated, varies with the type of genome and the type of viruses. The genomes of most DNA viruses are replicated in the nucleus, but some double-stranded DNA viruses are replicated in the cytoplasm. The genomes of most RNA viruses are replicated in the cytoplasm, but there are exceptions amongst the minus-strand RNA viruses. The retroviruses and pararetroviruses are special cases. Each replicates RNA to DNA in the cytoplasm and DNA to RNA in the nucleus. So again, there's a lot of variability, and we'll talk about the specific cases in detail as we go through the various virus families later in the course. The key enzymes used in virus genome replication are DNA polymerases and RNA polymerases. Many viruses encode their own polymerase, but some use a host cell enzyme. A DNA virus requires a DNA-dependent DNA polymerase, in other words, a polymerase that makes DNA from DNA. Amongst the DNA viruses that replicate in the nuclei of eukaryotic cells, viruses with small genomes use the cell enzyme, while viruses with large genomes, such as herpes virus, encode their own enzyme. Those DNA viruses that replicate in the cytoplasm must encode their own enzyme. The enzyme that replicates the genome of an RNA virus is often referred to as a replicase. For many RNA viruses, this is the same enzyme that's used for transcription. The retroviruses and the pararetroviruses, again, remember, they encode reverse transcriptases to transcribe RNA to DNA and use the host cell RNA polymerase II to transcribe from DNA to RNA. So this just shows you the types of enzymes that are there. So if you're making DNA from DNA, you use a DNA-dependent DNA polymerase. If you're making RNA from RNA, you use an RNA-dependent RNA polymerase. And if you're going from RNA to DNA, an RNA-dependent DNA polymerase or a reverse transcriptase is what you would need. So that's the various types of enzymes that a virus needs. So you see those up at the top, but if you look down at the bottom, there's only two types of cell enzymes that we see. Cells will make DNA-dependent DNA polymerases and DNA-dependent RNA polymerases, or RNA polymerase II. So if the virus requires any other kind of enzyme, it has to encode it itself. If it can use one of these two types of cellular enzymes, then it will. Assembly and release would be the next steps in virus replication. We said that assembly occurs from subunits, sort of akin to what we see happening in a car factory. A nice illustration of assembly of viruses is shown through phenotypic mixing experiments. Here's an example of a phenotypic mixing experiment. So you take two very closely related viruses. Virus A is indicated by blue, virus B is indicated by black. And you have the capsid proteins of each virus and you have the genome. Because these are two very closely related viruses, if you co-infect a cell with both viruses simultaneously, you can get a mixed infection. And when you look at the progeny that results from these mixed infections, you'll see that they're phenotypically mixed. And this is indicated by the different colors of capsid proteins inside the individual capsids. So on the right and the left, you have the parental type capsids, so all blue or all black. But the two types in the middle have a mix of virus A and virus B capsids. But if you take the virus, the second one from the left, for instance, that has the genome from virus B, but it has a mixed capsid, and then you grow that up, you do your second passage, the progeny virus are all going to be like virus B. And that's because this change was phenotypic, not genotypic. There was no interaction of the two genomes here. There was just a mixing of the capsid proteins during the assembly of the virus. And so that's what we call phenotypic mixing. But it's a nice illustration of how this happens in host cells as the virus is being assembled. The virus subunits and even partially assembled virus particles are often sequestered in particular portions of the cell, and these are called inclusion bodies. The inclusion body is a concentration of newly synthesized structural components into subcellular compartments. These are often seen in the late stage of infection of cells by many viruses. The inclusion bodies can contain viral replication complexes, transcriptional complexes, nucleocapsids, and so on, and they're accumulating in these specific locations in the cell. And these will form structures called virus factories or inclusion bodies. The location reflects the site of replication. So, for instance, herpes virus inclusion bodies often show up in the nucleus. And it's interesting because these particular structures are large enough that they can be visible in a light microscope. So normally you can't see virus using a light microscope. But when you have a virus-infected cell that's showing inclusion bodies, using the right stain, you can often see these in under the light microscope. So for instance, you can see up at the top there, those two structures that look almost like eyes. Those are inclusion bodies. They sometimes will refer to those as owl eyes. And I can remember when I was working as a research assistant at the University of Cincinnati Medical Center. I was working in a laboratory where they did kidney biopsies a lot. It was a kidney transplantation unit, and they did biopsies of the kidneys to check on the health of the transplant. And one of the big problems with a transplanted kidney was the development of CMV virus. CMV virus is ubiquitous and normally doesn't cause any problems for a person with an intact immune system. But when you receive an organ for transplantation, for the rest of your life you're given drugs to suppress your immune response so that you don't reject the organ. And sometimes in a patient who has received a transplanted organ and they become sick from other factors and their body's under stress, we'll see the upsurge of CMV virus in the kidneys of these patients. And I would just take a sample from from a kidney biopsy. My job required me to be out on the floor when they do biopsies. I take this sample and I do a frozen section, and I would just use toluidine B as the stain and look under the light microscope, and I could see these inclusion bodies. In these electron micrographs on the slide, over on the left you see cytoplasmic factories in a cell infected with reovirus, and on the right you see nuclear inclusions of herpes simplex virus 1. If you look closely, you'll see on that picture on the right, you can see filled triangles. Those are electron translucent nuclear inclusion areas. The filled arrows indicate full capsids. The unfilled arrows indicate empty capsids. And unfilled triangles show the host chromatin compressed to the periphery of the nucleus by the cytoplasm. If we look at this figure showing release, some viruses, as we've talked about before, are released by budding. You can see Newcastle virus buds from the plasma membrane. Influenza virus replicates in the nucleus, but it is then transported through the nuclear pore into the cytoplasm and buds from the plasma membrane. HIV is kind of similar to this, but then we go down to adenovirus and we see that it replicates completely in the nucleus and then is released by lysis. So poliovirus and adenoviruses, as in contrast to the viruses that bud through either internal membranes or through the plasma membrane, they actually are released through lysis. So here you can see lysis of a bacterial cell to release the progeny phage particles. Lysis requires the death of the cell, whereas remember, budding is a process that can occur over a prolonged period of time. And then, of course, envelope viruses are going to exit through budding, and we can look at this process in more detail. So we've looked at some pictures of this already, but envelope proteins will be translated by polysomes. So we have a process of synthesis and co-translational membrane insertion. So what this means is that the viral encoded protein is being translated by ribosomes that are attached to the rough ER. And as the protein is being translated, it's being inserted into the ER. Then glycosylation of the inserted portion of the protein can occur in the rough ER, which eventually is moved up to the Golgi apparatus. And this is how these viral glycoproteins get inserted into the plasma membrane. So the viral glycoproteins are transported to the plasma, the membrane in a vesicle, which then fuses with the plasma membrane. All right, so now if you go up to the top right, you have a situation where the viral envelope glycoproteins have now been inserted into the plasma membrane of the host cell. The nucleocapsid, which is formed during replication, will migrate to the virus-modified membrane, and you have interaction between the capsid proteins and the internal domain of these transmembrane virally encoded proteins. And what will happen is that more and more of these transmembrane proteins interact with the capsid, and it pulls the membrane around it and eventually you have full budding of the virus and free infectious virus. The last thing we want to talk about in terms of virus replication is transfection. Transfection is the infection of cells by nucleic acid alone. Now remember, the capsid and also the envelope of a virus particle is there in large part to protect the nucleic acid. Nucleic acid is easily degraded. If you remove nucleic acid from the capsid, the efficiency of infection is much lower because the nucleic acid is so easily degraded. In fact, the efficiency of infection is only about 1% compared to infection with intact virus. But you have the advantage that the host range is wider. Cells that are permissive to transfection may not be susceptible to infection by intact virus, and that's because receptors don't play a role in transfection. So as a nice example of this, consider chicken cells. Chicken cells are normally not susceptible to poliovirus. Poliovirus is normally grown in primate cells, and the reason for this is that chicken cells lack the appropriate receptor. Now you can use transfection to infect chicken cells, and the nucleic acid will initiate replication. But the virus particles that are released from the chicken cell cannot go on to infect other chicken cells because the receptor still is not there. So that is an important advantage to working with transfection in the laboratory. Also, nucleic acids are more resistant to heat than proteins are. The protein coat can be denatured, but nucleic acid is unaffected. This is particularly important to remember in vaccine production because it's possible to treat a vaccine virus in a way that denatures the protein coat but leaves the nucleic acid unaffected. This could be a disastrous result when you're making a vaccine because the virus particle could infect a cell and then establish a fully productive virus infection because the genome had not been affected. Another aspect of transfection is that nucleic acids are not affected by antiviral antibodies. Antibodies target proteins, so the proteins that are on the surface of the viral envelope or on the viral capsid will be the targets of antibodies, but nucleic acids will not be recognized. Another aspect of working with transfection is that in some RNA viruses, you can make a DNA copy of the viral RNA that is infectious. This can be really useful in the lab because RNA has a much higher mutation rate than DNA. Remember, DNA has a proofreading mechanism that RNA doesn't have. So, for instance, if you're trying to make a vaccine and you need to replicate the virus to very high levels, it can be useful to be working with a DNA transcript instead of an RNA genome because you can reduce the mutation rate. But these are aspects that are much more important in the laboratory as opposed to seeing what goes on with viruses in nature.